Good afternoon, everyone. Thank you for joining at this event from Veins to Bedside, the journey to creating a sight-saving drug. It is my pleasure to walk you through the conference and introduce you to all the speakers that have come here to share their knowledge. Today, early-stage researchers from the TransMed consortium, in collaboration with Retina International, will take us through a step-by-step process of drug development, clinical trials for the retinal diseases. Introducing myself, I'm Manisha, one of the early-stage researchers in the TransMed consortium, based in University of Iceland. Today, I will be co-hosting the event with my colleague Anya. We will have 3 sessions today. The first session will be about how the eye works and what retinal degeneration is. Meanwhile, the second and the third sessions will cover the steps of translation medicine from identifying drug targets to trailing them on patients. Each session will be followed by a Q&A where you will be allowed to ask the question. For that, you have to press Alt-Y. The chat section will be disabled for the accessibility reason. And then after Q&A, we will have a short break where you can stretch your legs or get a cup of tea or coffee maybe. Another important thing, the event will be recorded. So if you are not okay or uncomfortable with it, please log it off. And the recorded version of the event will be available in the Retina International page, and you can access it after the event. Before we get on to that, I would first like to welcome Christina Fasser for a few words. She is the former president of Retina International and has been an active patient advocate in fighting retinal disease for many years. Let's give a huge round of applause for Christina.
Hello. Thank you very much, Manisha. Can you hear me? I think so.
Yes.
Okay. Thank you very much. Thank you, Manisha. Thank you, Fiona. In the background, Fiona Waters, the Director of Membership Outreach and Communication, is organizing this first webinar, staging young researchers. The European Union has a wonderful program, the Marie Curie Klodowski program for young researchers that are doing a PhD degree. They are supposed to go into the industry. They are supposed to go to other countries. And it's a very challenging, however, very interesting program for young researchers. This program, the TransMed program, is the third in a row. Already had happened before. The first one was a program organized to identify molecules that would help to restore vision or to save vision in PDE6. The second one was to test these molecules and to go near to preclinical state. And this one is now one which is also looking how you can bring it best to the eye. These programs were led by Professor Backe-Durand, by Professor Pete Eckstein from Sweden, and Professor Marigo from Modena. And this is a very bright, and one of the things as we thought would be good is to bring it also to our membership, our young membership, because these young researchers are our future. They will look at development of new drugs, how to get it best and safely to the retina, and also to bring it to the clinical work. And these young ladies and gentlemen, they will be also those that will bring treatments to the market once, will work to help. And it's one of our most important things that we educate young people, get them interested in the retina, which is one of the most fascinating areas in research at the moment, I dare to say. And they will make sure that all these many still unsolved questions and secrets of the retina will be solved in the future. So I'm really looking forward to hear the young colleagues telling what they did in the last 1.5 years. The floor is yours. Thank you very much.
Thank you, Christina. You have been encouraging us throughout the TransMed journey. Thank you very much. Now, let's start our first session. The first session will be about what retinal degeneration is and what are its therapeutic challenges. Welcoming our first speaker for today is Meltem Kutluer. She is doing her PhD at University of Modena in Italy, within the consortium, of course. She studies how genes are different, how genes act differently in retinal cells that are dying. Today, she will help us take a closer look at the eye and the retinal degeneration.
Thank you very much, Manisha. Hello, everyone. Welcome to my talk. Today, I would like to start with a little story. When I was a kid, I asked my mom, why the sky is blue, trees are green, and my shoes are red? And she told me that because of the sunlight. Sunlight contains all colors. And after that, I learned that the sunlights come in all colors and it's dispersed in different way. When it hits the object, like my red shoes, and my red shoes observe all colors and just reflect one color, just red. After years, another question comes to my mind. Can all the people see in the same way? Before to answer this question, let's take a closer look how the eyes work. When the light comes to our eyes, it passes through to the tunnel window, which is the colorful part of our eyes. And the light is arranged here that we perceive. After that, these lights focused by the lens, and then lens project the light on the retina, which is located back of our eyes. The retina is the light-sensitive tissue. It can convert the light into electrical signals. And after that, transmit this light to the brain to create the visual perception. How retina can do this? It has two special cells, light-sensitive cells. These are rods and cones. Rods are responsible with dim light vision, such as to see in the night. An opposite way, cones are responsible with the well-lit condition and also the high acuity vision and the color perception, such as reading. Let's turn back to our first question. Can all the people see in the same way? The answer is no, because there are a lot of genetic mutations and also the retinal degenerations. And these affects the cell population in the retina, which is characterized by the losing of the cells. I would like to share my personal experience with you. When I was undergraduate student, one night, my brother recognized that I am having difficulty to see in the night. It was like recognizing the face of my friends or to have the difficulty to find the keyhole to open the door or climbing the stairs. Until that time, I didn't know that I had the difficulties because I was thinking all the people can see like me. After going to ophthalmologist, I was diagnosed with retinitis pigmentosa. That was the moment that I decided to focus my career on to do research about retinal degeneration, which is changing my life. Right now, as a doctoral student, one of the main problems that we are searching in the lab is the lack of the disease model to able to study with the retinal degeneration under controlled condition in the lab. What I'm trying to do is mimicking the disease with the cells as RP do. This will give us to better understanding about retinal degenerations and also how the eyes work. Nowadays, there are advanced molecular biology techniques, such as gene editing tools, bioinformatics, and others. And we believe that this kind of advanced tool will help us to solve the problems. Thank you very much for your time and attention. Stay safe, stay happy.
Thank you, Milton. Thank you for sharing your story with all the people here. The second talk will be given by Lee Wong. She is also a PhD student at the University of Modena in Italy. Her project is about drug testing for retinal degeneration. Today, she will talk about the special challenges researchers face when creating therapies for retinal diseases. Lee?
Yes, thank you, Manisha. So, hello everyone. Welcome to the second talk. And in this talk, we will briefly introduce some treatment therapies and challenges for retinal degeneration. So next slide, please. Retinal degeneration is a type of disease with a really big family. And inside this family, there are multiple known diseases. They can be really different in their pathology, prognosis, and visual symptoms. So it can be broadly divided into like inherited retinal degeneration that's usually caused by gene mutations. Like retinitis pigmentosa is the most common form. And others can be multifactorial retinal disorders. So this is complex, as many factors could influence the disease at the same time, like such as age, gene, or environment. Age-related macular degeneration is the most common form. Next slide, please. When we talk about this, of course, the treatment is always the first thing that we want to consider. So currently, there are some approaches under development, including like gene therapy, cell therapy, drug therapy, and others like retinal prosthesis. So for the gene therapy, this is currently the most advanced strategy for the disease, especially for inherited retinal degeneration. So it is using genes to treat diseases by delivering a gene into the cells instead of using drugs or surgery. There is already a first market approval gene therapy product for treating a retinal degeneration disease called type 2 label congenital amaurosis. Cell therapy, basically it's a cell transplantation strategy by replacing diseased or dysfunctioning cells with healthy and functioning ones. Drug therapy, so currently there are some quite promising drugs being identified in different retinal disorders, such as the use of anti-VEGF product in age-related macular degeneration and diabetic retinopathy is quite valuable. Some other approaches like retinal prosthesis. So those are some devices that are meant to partially restore useful vision for someone who already lost photoreceptors. So so far there have been some systems having received marketing approval for use in Europe or the United States. Next slide, please. Despite those quite promising strategies, we still face many challenges. The first is the heterogeneity of genes. Like inherited retinal degeneration, more than 270 genes have been identified. Such a complicated condition is not easy for developing a common treatment for a large number of patients. The second is that although it's now possible to identify the gene mutations in many forms of retinal degeneration and to predict their impact on cellular functions, much less is known about the mechanism that how they exactly cause retinal cell death. This also hampers the development of an accessible target. Even if a treatment has been developed like a potential drug or a gene, we need to deliver them into the right position in the retina. However, there are also some difficulties in delivery process. Like if you use eye drops or take orally, for those routes, they cannot support an adequate drug concentrations in the retina, as the retina is located in the really back of the eye, and also it is protected by some physical barriers. Of course, you can use some more efficient ways, like directly inject the drug into the space in the back of the eye called the vitreous cavity. But this approach for drug administration is applicable only in selected cases, for example, when treating age-related macular degeneration. Also, like gene therapy, it has a delivery limitation. So usually, genes are not easy to enter into the cells. So they need a vehicle to help them to enter. But currently, some vehicles have a size limitation. If the gene is too big, it couldn't be loaded onto the vehicle, then it couldn't be delivered properly. Another issue is that not every solution can be used in all stages of the disease. For example, like drug therapy or gene therapy, they're better to be used in the early stage when the retinal cells are still alive. So establishing the base timing and type of therapy is also very important and challenging because some patients may already miss the treatment window with most retinal cells like photoreceptors are already lost at the time of diagnosis. Okay, so and cost is also in the consideration too. So the costs for developing a gene or even mutation-specific approaches are quite substantial, while often having only a limited number of individuals that could potentially benefit from this. So, for example, like the current cost of a gene therapy product is very high and unaffordable for a majority of patients. Next slide, please. Finally, developing a treatment is not an easy task. It needs combined efforts. Scientists need to do cell research and animal research, and also it needs efforts from clinicians, from the patients, from some organizations, industrial, and so on. Only joined together, we can fight these devastating conditions. Thank you so much. Thank you.
Thank you so much. University student will close this session. In his project, he is looking for the genes and the proteins. They may have more direct detrimental effect in retinal degeneration. Today, he will be shortly introducing the TransMed program.
So, hello everyone. I'm Jiaming, now working in Lund University in Sweden. So before we go any further, I would like you to have a general impression what Transmedicine Consortium is and what we are doing right now. So next, I'm going to have a brief introduction about our program. Transmedicine is a network training program, and thanks to the European Union, they invest sufficient money to us for research purpose, and this grant is named after a very famous chemist, Marie Curie. The main purpose of this program is to train the translational scientists. That is because right now we actually have a lot of excellent scientists and scientific output. But unfortunately, the translation between this scientific output and the real world product are still very limited. For example, we would like some more devices that can make our life easier. We want some more medicine that can treat the disease. So this is the main aim of this program. It did not simply train the typical scientists who are going to find the truth of the world, but actually train the translational scientists who is going to make this work better. And in this program, we focus on the disease, the eye disease, retinitis pigmentosa, and our long-term goal is to develop the novel treatment towards this disease. To achieve this ambitious goal, a large collaboration with people with different expertise are required. And now we have 13 early-stage researchers who are taking part in this program. And next slide, please. Here is the general workflow of the Transmedicine Consortium. So although we have people with different expertise, we are not magicians. We cannot bring something from nothing. So at first, we have to have an idea what we are going to develop and bring this idea to basic research. We already know that one protein system called cyclic GMP PKG is very high in the retinal degeneration patient. So we consider this kind of the relationship between this protein system and the disease development. So we think that, is it possible that when we inhibit or block this protein system in the retina, it would be alleviate or even reverse the disease progress? That's our idea. So we are doing research in this area. For example, some of my colleagues in Germany, they will use an animal model that we know to have this retinal degeneration already, and then use the compound to treat the mice to see if this can elevate the depth of the photoreceptors of the retinal cells. And also some of my colleagues working in Italy, they have the material that can mimic the retinal cells of the photoreceptors and study the mechanism if we block this protein can elevate the cell depth of the photoreceptor. And also in this session, the first part, we would like to find out if there is any other proteins that are related to this protein system and have more direct detrimental effect in the retinal degeneration. Now, 7 biologists are taking part in this session. So in the first part, if the result is promising, we will go to the second part, making a drug. In this part, we have several pharmacists, chemists, and physicists working in this. For example, some of my colleagues in Iceland, they are working in the eye drop development formulation. And also in Sweden, we have some colleagues working in the compound development. But to be honest, even though we have the compound, we have the drug, that is not sufficient. We have to confirm that this drug can be delivered to the photoreceptor retinal cells very smoothly. So some of my colleagues and working to this part, we call that drug delivery system. And this is the summary of the second part, making a drug. And if this part is smooth, we can go to our final goal, go in clinic. We will organize a clinical trial to see if the drug we develop is safe, effective or not. And of course, we wish to make it commercialized and then to make it available to the people who need it. That is our long-term goal. To be honest, this is very challenging, but we are very excited to take part in this program. And next slide, please. So here are all, we are 13 early stage researchers. And then now, thank you for your attention. And please feel free to ask any question if you have. Thank you.
Thank you, Ming, for introducing our program and what are we actually doing in the consortium. This is the end of the talk for this sessions. So now the floor is open for questions. So if you have any questions, you can ask by raising your hand. For that, you can press Alt Y, so that will raise your hands. You can press Alt Y to raise your hands to ask the questions if you have any. We have a question from Eric Hartman. Maybe you can unmute it and ask the question.
Hello, Eric. If you would like to unmute yourself by
Hello?
pressing, there you are.
Okay, sorry about that. Had to find it on my screen. Thank you very much. It's very interesting. And I wanted to find out, are your researchers, these within the consortium, how are those grants awarded to these researchers? And how long do they last as far as getting other researchers in the consortium?
So thank you, Eric, for your questions. So to answer your question, I would like to say this program is sponsored by the Marie Curie grant. And then our supervisor, they will write this grant and then have this excellent idea. And then when this grant is approved, the money can be cover our salary and also for research purpose. This grant actually includes 4 years.
Thank you.
Yeah, I hope I have answered your questions.
Yes, thank you.
Thank you, Eric, for your questions. So if you have any other questions, you can raise your hand by pressing Alt-Y. I don't see any hands raising in the attendees. But Malcolm, I have a question for you. So you talk about this retinal pigmentation and what retinal degeneration is. So how important it is to know what kind of mutation you have as an RP patient?
I can answer this question as a personally. It's important because in the market, if there is any available gene therapy, and it will be the time saving because finding the mutations takes time. It's time consuming to do research about it. Of course, it will depend on the retina condition, and the correct answer will be given by your ophthalmologist.
Thank you, Milton. Let's see if we have any more questions from the attendees. I don't see any raising hands. Yes, but I still have one question for Lee as well. So you talked about all the treatment challenges in the RP. So how similar are these treatment challenges compared to other neurodegenerative diseases like Alzheimer's or maybe Parkinson's?
How you mean how they similar with?
Yeah, like how the treatment challenges are similar to other neurodegenerative diseases.
Similar is that well, for most of the, especially for the inherited retinal degeneration, so most of them are caused by gene mutations. So so the gene mutation has different types. It's really rather difficult to treat them. The second is that, well, I knew little about the Parkinson's disease, so I don't know how to compare them in both. But for me, like for retinal degeneration, the challenges are quite big because you need to target, especially for the retina, and the retina is, well, it's not easy to target this one because you cannot use like by surgery or by some other portraits, you can easily revise them. The degeneration of the retina, you can basically, for now it's only few approaches that you can treat them. Like the gene therapy now, it's only one product and it's only available. So we still have a long way to go for that.
Thank you, Lee. Let's see if we have any more questions from the audience. You can ask your question by raising hand. You can do that by pressing Alt-Y. I don't see anybody raising hands. Guess we don't have any more questions, but if you have any more question or if you want to ask later, you can also send an email to the Retina International page and we will ask the question to the relevant speakers. So this is the, if we have no more questions, this is the end of our first session. We will take a short break. Actually, we are slightly ahead of the schedule, so we will have a longer break than we expect. Thank you.
Good afternoon, my name is Anna Karpińska. I am your second host today, and I will be chairing this part of the event. Welcome to the second session of our conference. And it seems that there is one question from Eric Hartmann. So if you could please unmute yourself and ask the question to the speaker. Eric, could you ask the question? Your hand is raised.
Yes, I'm sorry. I was having a problem with the Word document download. When I download it, it is white writing on a white background, or inverted, it's just a black screen. So I do not see the content, though a screen reader does read that the content's there. I'm not sure what the formatting was. I just wanted to make you aware of that.
Oh, thank you. Ariana Tolon, PhD student from University of Tübingen in Germany, will start this part of our meeting. In her project, she is testing drugs for retinal degenerative conditions on mouse retinas. And today she will give us more insight on how and where to test the drugs.
Thank you, Anna, for this presentation. So as Anna said, I'm Mariana, and I'm going to talk about how and where to test drugs. Next, please. Next slide, please. Thank you. So what's the journey of a drug for an eye disease? In this seminar, we will learn how a drug is formed and which are the requirements for targeting what we want. But we don't know so much about this drug. And we need to test it for its efficacy, but we cannot test, of course, directly on patients, on people, because since we don't know so much, we don't know if it's causing some side effect, and we don't want to risk anybody's life. So there are several steps in between that a drug needs to perform before we can even think on testing or like giving to people. Next slide, please. So I said we need to test it for its efficacy. And the first model we can test our drug or several drugs that we produced is actually cell cultures. Cell culture is an extraordinary model because probably you don't know, but from the retina, we can extract special kind of cells. The cells are actually undifferentiated. So means that they didn't decide yet what they want to be. So we can extract them from a mouse retina. We can put on a sort of dish in a warm environment with special nutrients, and we can make them be what we want to be. And in this case, we want them to be photoreceptor-like cells. More extraordinary than that, we can make them starting to die. So like, for instance, in a retinal degenerative disease, like in retinitis pigmentosa. Next slide, please. Let's assume now that we have several drugs that we produce, and we want to test whether they are actually able to reduce the number of photoreceptor-like cells that are dying. So we have this dish containing cells with, we can make them starting to die, and we can answer two main questions with testing these drugs on cells. First question is, which of the number of drugs I have is the best? So which of these drugs is actually reducing the number of photoreceptor-like cells which are dying? The second question we can answer with that is, how much drug do we need actually to make the drug basically reducing the number of photoreceptor-like cells? Because sometimes the amount of drug can be too little with no effects, or too much with side effects. So now our drug, so we selected a number of drugs, or one drug that is actually very effective, is reducing the number of photoreceptor-like cells. And we need to test on something which is more close to the human retina, but of course not yet the human retina. Next slide, please. So for that, we will test our drug on organotypic retinal explant cultures. What are these organotypic retinal explant cultures? So now let's imagine that we can extract the eye from an animal model, let's say a mouse. We can remove the upper part of the eye and we will have a sort of cup. And in the inner part of the cup, there is the retina with all the photoreceptors and other layers of the retina. So now imagine that this cup we can cut in 4 points. The retina will resemble now a flower with 4 petals. And the retina will flatten, and then we can put on a sort of dish. In this dish, of course, we will give nutrients. We will put also nutrients to make it grow and survive, of course. And we will place the retina in a dish in a warm environment, which is resembling the body temperature. And here we can test our drug. Next slide, please. So what is special of these organotypic retinal explant cultures? As you remember before, when I was explaining about cell cultures, actually these are photoreceptor-like cells. So they are not real photoreceptors. But here we are testing actually our drug on real photoreceptors in a real tissue. Secondly, we extracted actually the eye from a mouse or whatever animal model with a mutation which is resembling the human situation, so a human disease like retinitis pigmentosa. But how do we check here if our drug is effective or not? So we can collect different kinds of retinas, so different retinas from the animal that we are using here, and basically one part of the retina, one group of the retinas we collected, we can treat with our drug, and the other part will be not treated, untreated. So now we can label our cells which are dying. And how do we do that? So actually there is a substance which is able to recognize the dying cells and emit fluorescence under the microscope. And how does it work? So basically imagine that the cell is a sort of balloon. And if we pour a glass of water in the balloon, no water will enter the balloon, of course. But if we make a hole into the balloon will lose its shape and there will be a hole. So what we can have is basically that if we pour the glass of water on the balloon, some water will enter the balloon. And this is how basically the substances work. So it's penetrating the disrupted cells and basically it's recognizing something that make the cell like dying, and it's emitting fluorescence. Now that we labeled our dead cells, we can count the number of photoreceptor cells which are dying and compare. Is our drug actually reducing the number of photoreceptor cells which are dying? If the answer is yes, then we can move to the next step. Next slide, please. So now we need to test our drug on a living animal, in a living body. And so we will administrate our drug, for instance, on a mouse first, or another kind of rodents, which is a small animal. But then we need to administrate our drug on an animal which is closer to human, for instance, like a monkey. This steps is very important because will give us information on if the drug is actually producing any side effect on the body. And it's very important to make our drug ready for the next step, which is actually towards the clinical trials, which will be explained later by my colleagues. Next slide, please. With this, I would like to thank you for your attention. And if there are any questions, of course, I will be very happy to answer.
Thank you so much, Ariana. Our next speaker is Michelle Rasmussen, a PhD student from Lund University in Sweden. The goal of her project is to find new proteins that might help to explain the retinal degeneration and potentially become new therapeutic drugs. In her talk today, she's going to tell us definitely more about that.
Thank you, Anya, for the presentation. So my title of my presentation is called The Journey of Discovering New Potential Therapeutic Targets in the Retina. Next slide, please. So why do we need new targets, and what are these targets? These targets can be proteins, and proteins are the building blocks of our body. They can be involved in building things or breaking down things in the body. If these proteins are involved in, for example, retinal degeneration and have an important role in this degeneration, they can help us understand the retinal degeneration that, for example, occurs in retinitis pigmentosa. And then they can also become new therapeutic targets in the future. Next slide, please. So some of these targets that I'm looking into is proteins that can bind to cyclin GMP. Cyclin GMP is a tiny molecule that exists in all our organs in the body and also in the retina. We have seen that the cyclin GMP level is increasing during retinal degeneration in the retinitis pigmentosa. And therefore, cyclin GMP is a very nice marker for retinal degeneration. But then the proteins that may bind to cyclin GMP may also be very relevant markers and maybe therapeutic targets in the future. Next slide, please. Finding these targets is like fishing. So therefore, I have on this slide a guy who's fishing. He has a fishing pole and a bait, and then he has specifically chosen also the area he wants to fish in, if it's in the ocean or in the river, because he wants to catch a specific fish maybe. I'm using the same principles when I'm looking for targets. Here, my fishing rod and my bait is the method and the bait in my experiments, and the river or the ocean is the tissue I choose to so-called fish in to find my proteins of interest. Next slide, please. So I asked myself 3 questions when I have to generate a protocol to find these targets. Question 1, what tissue should I use? Of course, I'm interested in the retina, so I will use the retina. But the retina, there's different areas that could be interesting to look in. Or there are also different mouse models that has mutations that resemble retinitis pigmentosa. So should I use one of these mouse models or more of those? And should I also use a healthy mouse model? Next slide, please. My question 2 is, what protein should I look for? Of course, I'm looking for proteins, new proteins that have never been shown before to bind to cyclin GMP. But in the literature, I also know that there are some proteins that have shown to bind to cyclin GMP. And these proteins will be my so-called positive controls. So I know that my method of choice and my experimental protocol works. Next question, slide, please. Question 3, how do I find these proteins? What method should I use? I have to think about that I'm only interested in the proteins that bind to cyclin GMP, and also that the retina is very small. So I have to think about these things when I design the protocol and what method I choose. Next slide, please. So for my experiment, I chose the entire retina as my tissue. I did this because I need a lot of retina, a lot of sample in my method. And also I chose 4 different mouse models. I chose a healthy mouse model, and then I chose 3 mouse models that had mutations that resemble retinitis pigmentosa, so I could compare the outcome of my results. Next slide, please. For the bait, I chose, of course, cyclin GMP, since I'm interested in the proteins that bind to cyclin GMP. But the cyclin GMP molecule, in my case, had to be modified a bit, meaning that it was actually bound to a tiny, tiny, tiny ball that you can barely see. And this was then my bait. Next slide, please. This bait I used together with the method called affinity chromatography. And affinity chromatography is a very simple method, actually. Basically, you take your bait, and here you add your retina, which is in solution. And then the idea is that all the proteins that bind to cyclin GMP will bind, and then you can wash away all the other proteins that are not of your interest. And then you will have, in the end, cyclin GMP and proteins of interest. Next slide, please. But then the question is, how can I identify all these proteins remaining in my sample? where I have cyclin GMP and the proteins. And here I use a method called mass spectrometry. And this method basically goes in and identify all the proteins that I had left and gave me a list. And in my case, it gave me a list of 400 proteins, for example, and that's way too many proteins for me to go in and investigate. So I had to use the literature and criteria to scale this list down. And I ended up with 20 proteins, where half of them were proteins that I knew bound to cyclin GMP, meaning they were my positive control that verified that my method worked. And then the other half were proteins that may bind to cyclin GMP and thus be relevant for retinal degeneration. And of course, then these proteins has to be further verified if they really bind to cyclin GMP, and also if they are relevant for retinal degeneration and could be therapeutic targets. And this is what Irena previously talked about, doing cell cultures or on retinal explants. Next slide, please. Yeah. So this was very shortly the journey of discovering new potential therapeutic targets in the retina. Thank you very much.
Thank you so much. I am happy to welcome our next speaker, Akansha Roy. She is a PhD student working in PAMGym in Netherlands, and her study is focused on identification of new proteins that could be causing retinal degeneration. And today her talk is going to bring us closer to the biomarkers in retinal disease.
Thank you, Anna. Hello, everyone. Today I will tell you about biomarkers in ocular, that is, eye diseases. Next slide, please. So the first question that comes to our mind is, what exactly are biomarkers? When we are sick, we go to a doctor. The doctor asks us to go for medical tests. And based on the results, the doctor confirms if we are sick or we are healthy and free to go. Therefore, biomarkers are indicators of a disease, and the doctor arrives at the conclusion by looking at biomarkers. Next slide, please. So now the next question that comes to the mind is, where exactly are these biomarkers present? Biomarkers are present in the samples which the patients provide to the doctors. The samples can be blood samples, or they can be from body fluids like saliva samples. Saliva samples are also very common nowadays to test for coronavirus symptoms. The test samples can sometimes also be part of the skin to check for certain type of cancers. In the eye, the biomarkers are present in the test samples, which can be tear samples or the fluids which are present inside the eye. Next slide, please. So now the question that comes is that what exactly is importance of biomarkers? Biomarkers are very important in every stage of drug development, right from the very initial stage when a new drug candidate is discovered. The effect of the drug candidate there is measured and studied on specific biomarker. During the drug development phase also, where the drug is changed and is designed, there also the efficiency of the drug is tested every time on its effect on specific biomarkers. In the end, the confirmed or the validated biomarkers, they serve as endpoints for clinical trial. Next slide, please. So now I will briefly tell you about my PhD project. The aim of my PhD project is to identify new biomarkers for retinal degenerative diseases. For this, of course, first I need test samples. So I use cells which are derived from a part of the eye called retina, or I also use retina tissue. I add these test samples to a chip. You can think of a chip as a TV remote, but with only 4 big buttons. But here, all the 4 big buttons also have 142 very, very small buttons. So the test samples, the things which are present in the test samples, sometimes they bind to any of those 142 small buttons. Wherever a binding takes place, we see a bright white spot. And where there is no binding, there I see a dark spot. So my job is to analyze those black or white spots, and in the end, I identify some potential small buttons or small spots which can serve in future as biomarker for retinal degenerative diseases. So now finally, to summarize my talk, biomarkers are indicators of any kind of disease. The biomarkers are measured to identify the stage of the disease, the type of treatment to go for, and to also monitor the efficiency and response to the treatment. Thank you so much for your attention, and any question?
Thank you so much, Akansha.
Thank you, Anya.
Eriko Himawan and his talk will actually close this session. And Eriko is PhD student working in INOCURE in Czech Republic, and he is currently working on development of drug delivery system for treatment of retinal disease. His talk will introduce us to those approaches.
Hi, my name is Eriko Himawan, and it's a pleasure to be here with all of you. And today I will talk about drug delivery system. And my talk is the first part of this topic, which is to introduce you to vehicles for drug delivery, for drug to reach its destination. Next slide, please. There are 3 things that I would like to share with you today. The first is, why do we need drug delivery system? Second is how they bring benefits to formulation scientists and to you as patients. And the third is, what are the examples of these drug delivery systems? Next. Let's go with the first one. Why do we need drug delivery system? The key word is delivery. Now, when you think about delivery, regardless of delivering a love letter, a package, a Christmas present, delivering a person, or a drug molecules, in the process of delivery, there is always movement involved. So movement involved from point A to B. Next. And why is the movement then important? So now imagine you want to have a Christmas holiday in Prague. Depending on where you are, you can drive a car, ride a train, or fly in an airplane. All this transportation system can deliver you to your holiday destinations. Now, this is the reasons why we need drug delivery system. It's the same with drug molecules. You use special vehicles like car, train, or airplane to help it reach its destinations. So we need this to help with the movement. Next. But how does this drug delivery system actually bring benefit to formulation scientists and to you patients? It's very simple. Imagine again that you are going to Prague for a Christmas holiday, but this time you are walking. Doesn't matter where you are, doesn't matter if you are in China, doesn't matter if you are from Germany, doesn't matter if you are from France, you have to walk. No car, no train, no airplane. What do you think will happen? Now, unless you are very close to the border of Prague, or unless you are a Superman, you may not reach it. Now, using the same analogy with drug, if you use drug without a proper drug delivery system, it is the same with walking instead of riding a vehicle to reach its destinations. It is possible, but it may cost you too much time, too much energy, and still have a high chance of failure. Next. Now, I would like to emphasize again on the benefit. With a proper delivery system, a formulation scientist like me can help drug reach target faster and in more efficient way. And because of this, it can bring more benefit to the patients. Why? Because this may lead to higher chance for treatment to be successful. If the target can be reached faster, and if the drug can be uptaken more efficiently, in many cases, it will help in increasing the efficiency. And because it helps in increasing efficiency, it may also help reduce the cost of the treatment. How is it possible? Imagine before, if you just use a normal drug without proper vehicles, without proper delivery system, you need 100 milligram of dose each time you do the treatment. But with the proper vehicles, with using car, train, or airplane, now you just need 1 milligram instead of 100. So this may help reduce the cost a lot. Next. Those are the concept of why it's important and how it brings benefit. The last topic that I'm going to share with you is what are the examples of these vehicles for drug. Now, just like there are many types of vehicles to help you travel to Prague for your holiday, there are many types of vehicles that can help drug reach its target. Some example of these vehicles includes, but not limited to, liposomes, solid lipid nanoparticles, and cyclodextrin nanoparticles. So this term may sound scary to you, but don't worry. If at one point in the future you hear someone talking about this and they mention drug delivery system, just imagine that they are basically car, train, or airplane. The last thing that I want to highlight here is that there is no such thing as perfect transportation system. One type of transportation system may be better than the others. For example, if you're from China and you want to go to Prague, it doesn't make sense to use car. So airplane would deliver you to your destination faster. The same thing if you live in the border between Germany and Prague, using train could be better than using an airplane. So there is no absolute best transportation system. The same thing can be applied with drug. There is no type which is much better than the others. So it depends on the situations and it depends on the target. Next. I would like to conclude this talk with 3 things that I shared with you. The first one is why it's important, because it helps the movement of the drug. The second is how it brings benefits, because it helps the drug reach target faster and in more efficient way. And because of this, it may help increase the chance of the treatment to be successful and more cost effective. And finally, some examples for the drug vehicles. If you ever heard about the term drug delivery system, just remember, they are basically transportation system like car, like train, like airplane, but for drug molecules. Thank you.
Thank you so much, Enrico. The presentations in this session are over, so if you have any question, please raise your hand. So if I have a question to you, Enrico.
Yes.
During development of the drug delivery system, are you facing any barrier? For instance, some biological barrier, if we are talking about the drug delivery system for retina?
Oh yes, there are a lot of barriers actually. Retina is one of the most difficult target other than brain. because in retina, you have several barriers that you have to overcome. And unlike other organ in the body, you cannot just open the eye and sew it back. So it's very difficult. And one of the barriers that has to be taken care of during formulation, Anna, is what we call as vitreous. Because when you deliver drug to the vitreous, the vehicles have to be able to move to reach retina. So this sounds simple, but actually there's so much barrier in the eye, and many formulation fail because they just can't reach the target. So yes, it's a challenging, but here we are, TSR, trying to do our best.
So how are you actually overcoming those barriers from the chemical point of view?
All right. So like many different transportation systems, let's say car, there is normal car and there are Ferrari, right? So different types, although they're, car. What we are doing here in TransMed is that we are trying to make Ferrari for our drug molecules. So we are trying to make it as best as possible. For example, to make some coatings on the surface so that it can help move faster in the vitreous. So this is one thing that is being considered. Another thing is to make our vehicles, our car, to have more space, so there are more drugs inside. In this case, when the car reaches the target, which is retina, it can deliver more loads, and so with the hope that it will bring more benefits.
Thank you so much, Erico. We have one question from Eric Hartman. So Eric, if you could please unmute yourself and ask your question.
Yes, thank you. My question had to do with the creation of mouse models. My foundation is the Choroideremia Research Foundation, and 20 years ago, it was very difficult to create a mouse model. We did get one, but it was a knockoff model that used a woodchuck hepatitis virus, which made it hard to transport or to be used in different animal houses. What type of gene editing these days is making it easier for the creation of better and more accessible mouse models?
I assume this question is to Ariana.
Sorry, thank you for the question. So I'm actually not aware about the gene editing method that are used for this mouse model, so I'm sorry for that. So what I can say is the mouse model that we are using, so we are using here. So for instance, we are using the RD1 mouse model specifically for the retinal explant cultures, which is actually what I'm working on for my PhD. Which has basically 2 mutations, and this is our spontaneous mutation. So it was kind of very lucky founded, and it's actually having a sort of a very severe degeneration. So it's a model very characterized because it's long time back we have this model, but there are also other models that instead we use indeed we specifically mutated the genes. So the genes have been specifically mutated, like for instance the RD10 mouse model, which is also a model for retinitis pigmentosa, like the RD1, or other kind of models that instead are mimicking the cone dystrophy. And there are several mouse model available for the retinal degeneration for both Rhodes and cone dystrophies. So, but the kind of gene editing, I'm not really aware how it's done, so I'm sorry for that.
Thank you so much, Ariana, for your answer.
I hope I answer well for him.
There is also another question from Osvaldo. Could you please unmute yourself, Osvaldo, and ask the question?
Yes, can you hear me?
Yes, perfectly.
Okay, so mine was a little bit about the previous question about the targets, the barriers to reach the targets that Eriko was talking about. I just wanted to give an example. So for example, the vitreous that Eriko was talking about, which is a liquid inside the eye, is interesting in that it's moving forwards towards the front of the eye. So if you're applying a drug that you're trying to reach the retina with a drug, you're applying that drug in the front of the eye, you're actually fighting against the flow of this liquid, of the vitreous. So that's one interesting one. Another one is the sclera of the eye, or the white of your eye, is made of several different layers. So if you're trying to use eye drops, they are actually more complex than you might think because of that.
Thank you so much, Osvaldo, for your comment. So is there any question? If so, please raise your hand. So if I can, I have actually a question to Michelle about the protein. How you actually, once you find the suitable protein, what's happening later on? Well, how the further development looks like?
Yes, to make it simple, when I look into a protein, I test the expression of the proteins in the retina. If it's more or less expressed in a retinal degeneration mouse model compared to a healthy mouse model. And then if I found something interesting there, a difference, the next step is to do cultures on retinal sections, with like Ariana presented. And here I will try to inhibit or activate the protein to see if it could prevent cell death in the retina. These are the next step for me to test.
And how long actually it takes to find the suitable protein?
For me, it took around 2 years to find some of these proteins, because I had to collect the retina from the mice, which took a very long time. And for testing later on, if they are interesting, these proteins, it can also take a very long time because I'm depending on mice. So yes, that could take a very long time.
So it's also very challenging, I assume.
Yes, finding the targets is very challenging because you have to include the right criteria and read in the literature which is most reasonable. And then for the next experiment, you have to find compounds where you can actually test these proteins if they're relevant or not. And not all of these proteins are maybe produced in the labs, so I can't really test them that easily. So there are some challenges there.
Yeah, so if they can't be produced in the lab, I mean, how to deal with the problem?
Well, one is to take another protein of interest, because I'm not able to produce the proteins or the activators or the inhibitors. Or it is to start a collaboration with people who can generate these activators or inhibitors, and we have one of these in our group, in the consortium. But they may also be very time consuming for them, because they also have to verify if their product works in the end. So that could also take years, I think.
Thank you so much.
You're welcome.
So is there any other question from the audience? So if I can ask, I have another question to Akanksha, because I'm just wondering if it's possible that one biomarker can detect more than one disease, and how you actually overcome this kind of problem.
It's a good question. So yes, so one biomarker can also be the same for some other kind of disease. But right now my focus is on retinal degeneration. But since retinal degeneration is a type of neurodegenerative disease, So we also have other kind of neurodegenerative diseases like Alzheimer's. So it's very much possible that the targets that I find, they might also suit some other kind of neurodegenerative disease. And if that's the case, then I think it's a good finding. But right now my focus is just on retinal degenerative diseases.
Thank you so much, Akansha. So is there any other question? If not, maybe it's a good time for a little break. So let's say 10 minutes. So we can meet 1:30.
Welcome back, everyone. This is the last session for today. The session will be about drug delivery system and clinical trials. Our first speaker for this session is Osvaldo Perez. He is a PhD student at Research Institute of Sweden in Södertälje, Sweden. His PhD project is about finding the safest and most efficient ways to make drugs in larger amounts. In his talk, he will explain what a drug is and the role of chemists in drug development.
Okay, can you hear me?
Yes.
Okay, thank you for the nice introduction, Manisha. Hello, everyone. I'm Osvaldo, and in our group, I take the role of a chemist, specifically a process chemist. And in the picture on the right, you can see me in full lab gear in front of a large reactor, chemical reactor, ready to do some chemistry. And I will explain exactly what I do in a little bit more detail in a few moments. But first, I wanted to, next slide, please. Thank you. First, I wanted to take a moment to look back at what we have learned so far. We have a little spoiler there, but no worries. We've had talks about the retina, inherited retinal degenerations like retinitis pigmentosa, and the challenges in treating them. We've also had some talks about how to target this disease with drugs, how to test these drugs, and how to help the drugs reach their target. And in all of this, I feel like there's a central thing that we haven't talked about, and that is right there on the center of the slide, that is the drug. So all of this revolves around the drug. You know, we're trying to discover this drug that has the perfect effect. We're trying to make sure it works and trying to help it do its job as best as possible. And so as a chemist, I thought I would tell you what a drug is, since we haven't really talked about that, have we? So what is a drug? So the definition of a drug is a substance that causes a biological effect. So where Eriko was talking about drug delivery system as the vehicle, the drug can then be thought of as the driver, right? Because a vehicle doesn't really do anything without the driver. So, and a drug is, so a drug causes a biological effect that can be used to treat, cure, prevent, or diagnose a disease. But what is it really? So a molecule really is basically a molecule. A drug is a molecule with a unique structure. Next animation, please. In nature, structure is function. Okay, just like in your kitchen, your cup of tea gives it, the shape of your cup of tea gives it its function. It's the same in the molecular level. So I thought I would show you our drug. So it's right there in the yellow box at the center of the screen. That's the structure of what we are working with. And to you, it probably just looks like a collection of pentagons and hexagons, you know, joined together. But I can tell you that particular structure has a very important biological effect for us, which is, as Ariana explained before, that it prolongs the life of photoreceptor cells. So all of this was discovered by people that came before me, you know, hugely talented people. And they basically discovered this drug, and they came to me and they told me, Osvaldo, we have this great drug candidate, has an amazing effect, and we want to try to develop it as a drug. But we only have a few milligrams of it, and we are going to need kilograms. So please, we need you as a process chemist to develop a process to make more of this drug. Okay, next slide, please. So that's what I do. I do process development. And process is just a method to prepare this drug by chemical synthesis. So it's just a recipe, basically. So my job, in short, is to find the best possible recipe that we can use to manufacture this drug in larger amounts. Okay, so such a process needs to be many things, but mainly two things. One is that it should be scalable, okay? That means that you need to be able to do the same procedure that I do, just bigger, larger equipment, bigger amounts of material, and it should work. So just like these Matryoshka dolls on the right-hand side of the slide, they look the same, but they get bigger and bigger. It needs to be scalable, of course, because as I said, we're going to need thousands of kilograms of this drug eventually in order to meet demand if it is successful in clinical trials. Secondly, next slide, please. This process should be safe because actual people are going to be handling tons of chemicals in order to manufacture this drug. So that needs to be safe. So these demands, they're simple enough to think about, but they are more difficult than it seems to meet. They're quite difficult to meet. So to illustrate this, I wanted to take an example of a process that we all know, hopefully, straight from the kitchen. Okay, so next slide, please. Let's say that we are cooking pasta. That's easy enough in the kitchen. Okay, you just take a pot, you fill it with water, put it on a stove, and when the water is boiling, you just throw in some pasta and you stir it around a bunch, and then voila, you have your pasta. Okay, so that's pretty easy, right? But is that a good process? Is it scalable and is it safe? Let you think about that for a second. So in order to find out, let's do a thought experiment. Let's imagine that we try to do the same procedure you just do in the kitchen, but bigger. So let's say that our pot of water is now not 10 centimeters wide, but 10 meters wide. You blow it up to the house of a house. Immediately, you'll see that you will run into some problems. For example, problem number one, where do you find such large equipment? Okay, you're going to need a really big spoon, for example, to stir that much pasta, and IKEA is not going to help you with that, even here in Sweden. And if you do find your spoon, it's going to be still too big for you to move. I mean, you're going to need super strength, right? I mean, where do you even find that much pasta? How are you going to get up there and manage to throw in so much pasta anyways? So it's really not very practical, is it? Okay, so let's say that you're friends with the CEO of AstraZeneca and he helps you out and you solve all those problems. Well, you're still going to need to get up there to stir your pot of pasta, right? So you're going to need to stand on top of a huge pot of boiling water to stir it. And so, of course, you're for sure going to get burned by the steam. And also, what happens if the water starts boiling over, bubbling over the edge of the pot, as it happens to me embarrassingly many amounts of time. So that's easy to solve in the kitchen, where you can just reduce the heat and clean up with some tissues. But if your pot is the size of a house, then you have a real mess, so it's not a safe process either. So suddenly you have something as innocent as cooking pasta, which it seems to really not be a good process for a process chemist. So those are the exact same kind of challenges that we face in drug development, the same challenges. But we are trained for that. And eventually we do find a good process, which is scalable and safe. And then we can manufacture our drug in much larger amounts, as I have done. And we can provide that material to our colleagues so that they can continue doing their magic and developing a treatment out of this drug. So that's what I do. Next slide, please. That's what I do in front of that chemical reactor, what I'm trying to do. Thank you.
Thank you, Osvaldo. It was very interesting. I would like to introduce our next speaker for this session, Dilip Urumi. He is also a PhD student at Research Institute of Sweden, where he is developing new methods to carry drugs where they need to go when treating retinal diseases. He will be talking about the drug delivery system and the drug administration.
Can you hear me?
Yes.
Yeah, thank you, Manisha. Thank you, Manisha, for the introduction. Hello, everyone. I'm Dilip Urumi. I assume that now you have some understanding about what is drug and what is drug delivery system. So let's talk about how a drug's administered. Next slide, please. Firstly, what is a route of administration? So simply, it's a way the drugs are introduced into the body. As an example, you take tablets or capsules or syrups orally by mouth, which is one route of administration. You take injections into different parts of body or apply ointments onto the skin, which are other routes of administrations. Likewise, there are so many routes by which the drugs can be introduced into the body. Next slide, please. So why we need different routes? Yes, we need different routes because every route has its own purpose. And there is no common route of administration that can be applied for treating every disease, because every disease needs to be treated in its own way. And it's very important to select the best route out of the available options. So how do we select the best route? There are so many factors that we need to consider for a selection. And these factors are interrelated to each other. Some of the important factors are body site, type and severity of a disease, patient comfort, and type of drug and drug formulation. So we talk about a few examples here. Let's say you have a skin rash. How do you treat the skin rash? The best option is to use an ointment because the skin rash is a skin disease, not the whole body disease. That's why we go with an ointment. But what happens if the same disease, the same skin rash continues to grow all over the body? Can you still use an ointment? The answer is no, because the severity is high. That's why you see it's all over the body. So then you have to go with a tablet or a capsule or an injection based on the severity. So this is one example. The other example is a fever. So can you use an ointment in case of fever? No, because the target is the whole body, not the skin. That's why you cannot use an ointment. You need to take a tablet or injection based on its severity. That's why target body site and severity of disease are very important to consider. We'll talk about the other factors in the next slide. Let's assume we want to deliver the drug to the retina. Next slide, please. Let's assume we want to deliver the drug to the retina. On the left-hand side of the slide, you can see an eye with possible routes of administrations. We want to deliver the drug to the retina, which is sitting at the back of the eye, which is in dark blue color at the back. We have three options to deliver the drug here. One is to go with a routine injection to the arm. The other one is eye drops, and the next one is injections directly into the eye. So firstly, routine injection to the arm may not work in most cases. Why? Because here the drug enters the blood and then it has to cross so many barriers or obstacles before it can reach retina. And by the time it reaches retina, the drug may get ineffective and it may not be in a sufficient concentration to treat a disease. That's why the first option is not so recommended. The other option is eye drops, which are nothing but the ones you apply on the surface of eye. Even here, the drug needs to cross few barriers before it can reach retina. But the eye drops are very convenient. So the selection of eye drops is mainly depends on the properties. I mean, the type of drug and the formulation. Let's say if the drug can reach those barriers, if the drug can cross those barriers and reach retina, then we can use eye drops. In most of the retinal degenerations or retinal conditions, eye drops may not work because drug properties will not allow to use eye drops in most of the cases. So the best option here is to use injections because the injections can deliver the drug close to the retina. And in this case, the number of barriers for the drug to phase or very, very low, and the drug can readily be available at the surface of retina. So that's why the best possible route of administration to the eye, especially for retinal degeneration, is injections. But here there is a problem. Injections cannot be taken very frequently, like few injections a month are also difficult in case of eye because of poor patient comfort. But in case of complex diseases like retinal degeneration, you still need to go for an injection because the requirement is to treat the disease and the patient comfort comes later always. So that's why comfort may be compromised by the requirement in case of retinal degenerations. So what are we trying to do in TransMed is that we are making formulations. We are trying to make formulations that can either be given as eye drops or injections that can treat retinal degenerations. Next slide, please. Thank you.
Thank you, Dilip. Our next speaker is Sumaya Belhaj, PhD student from University of Tübingen. She's currently working on development of biomarkers for retinal degeneration. And today she's going to introduce us to the history of clinical trials and what happens to prepare drugs for clinical trials.
Thank you, Anna. So this is the final part of this webinar, and together we are going to go through the last step before a drug is reaching the market and the patients. And this last step are the clinical trials. So next slide, please. So first we are starting with a definition. According to the World Health Organization, the clinical trials are a type of research that studies new tests and treatments and evaluates their effects on human health outcomes. And before walking you through how clinical trials are conducted today, I would like to take you through a small journey back in time. Yes. And our journey starts in 1747, and this is the year the first systematic clinical trial was conducted by James Lind. James Lind was a surgeon on a ship, and on this ship there was a high mortality of sailors because of scurvy. So now we know that scurvy is caused by a deficiency of vitamin C, but back then they didn't know. So what James Lind did is that he selected 12 sailors who had similar symptoms, and he distributed them in 6 groups of 2 sailors. Each group was testing a potential treatment. So the first group here of 2 sailors, of course, all the sailors were under the same diet. The first group was testing additionally to its diet some cider. The second group was testing 25 drops of elixir of vitriol or sulfuric acid. The third group was testing 6 spoonfuls of vinegar. Group number 4 was having half a pint of seawater. The fifth group had 2 oranges and 1 lemon. And the last group was having a spicy paste with barley water. So after 6 days, they ran out of oranges and lemon, but it didn't matter because the 2 sailors under that treatment recovered completely. Also, James Lind observed an improvement in the group of sailors receiving cider. So now, as you can imagine, it's not as simple today to conduct a clinical trial. And now I would like to walk you through all the steps that led to the current clinical trials and how they are conducted now. So about 1 century after James Lind, another very important concept and a milestone in the history of modern clinical trials appeared. And this is a placebo. A placebo, to make it easy, is similar to the treatment but doesn't contain the active substance. So typically, if the treatment is a pill, the placebo would be a similar pill but made out of sugar, so not having any effect, for example. And the people receiving placebo are a control group. So you can know for sure that the group under the treatment and the effects observed in that group are due to the active substance and not to a random effect. Okay, 1 century after that, there are 3 very important concepts in clinical trials that emerged. The first one is a random assignment of individuals to different groups for the experiment, and this is called randomization. And if you relate to the James Lind clinical trial, the first one, he did already that because he took 12 sailors and he distributed them randomly into groups of 2, and each group was receiving a specific treatment. The second very important concept is replication. Replication is means just that measurements need to be replicated, so it should be repeated. And this is very important to make sure that any effect that you see is certain, so it's reducing uncertainty. This concept of replication is very important in science in general. So if you relate again to James Lind and his first clinical trial, to be sure that the effect of the orange and lemons were true, and we were certain that they are curing scurvy, then this clinical trial and this setting should be repeated again with a different group of sailors. The last concept is blocking. Blocking is a fact of arranging experimental groups that are similar to each other. And this concept was met in the first clinical trial by James Lind because he took, if you remember, a group of 12 sailors that had similar symptoms, same diet. So all the effect that would be seen would be due, hopefully, to the treatment. Next, please. And then after all these concepts were set, the first randomized trial was conducted in 1946-1947 in the UK at the Medical Research Council, and it aimed at testing streptomycin for pulmonary tuberculosis. And this trial was placebo-controlled, randomized, and also double-blind. Double-blind simply means that when the patients or the individuals are shared between the groups randomly, as we said, neither the patients nor the medical staff knows if a patient has a placebo or has a real treatment. So now that I walk you through some methodological concepts, I would like to talk about the ethical and regulatory framework of the clinical trials. Next, please. So the origin of the ethical framework is taking it's the Hippocratic Oath. This oath is specifying the prime duty of a physician, which is to avoid harming the patients. Unfortunately, this was not much respected in human experimentation, and most advances in the protection of human subjects were a response to abuses that were done, for example, during the Second World War. And this is when the first international guidance on ethics for medical research involving human subjects was born. And this is a Nuremberg Code in 1947. And the Nuremberg Code is highlighting the essentiality of informed consent. So this means that every subject taking part in a study should give its consent to it. And this consent should be given after all the study, the risk, and how the study is going to be performed are explained to the patient. That's why it's informed. In parallel to these ethical guidelines, the government authorities started recognizing a need to regulate and controlling these medical therapies. And this is how the Food and Drug Administration was born in the US in 1862. Please, next. The equivalent of the Food and Drug Administration is in Europe is a European Medicines Agency, and it was funded in 1995. And these two organizations are the ones responsible to give the approval for any treatment or drug to be delivered to the patients in their own territory. Okay, so now we saw the methodology, we saw the ethical and regulatory framework, and now we would like to walk you through the different phases of clinical trials. And we are going to start with the preclinical phase. Next, please. So this preclinical phase is done on animals, and it's aiming at testing the drug. Next, please. At testing the drug safety in doses that are more or less equivalent to what the human subjects are going to be exposed to. So first, the drug should be safe. Then also this preclinical phase is exploring how the drug is processed by the organism and how the drug is exerting its effect on the organism. So these two different things are named pharmacodynamics and pharmacokinetics in scientific terms. Next, please. And all the data or the results collected in this phase are very important because they need to be submitted for approval if the drug is to be further tested in human subjects. And the testing of the drug in human subjects will be explained by my colleague Laura. I thank you for your attention and I would be happy to take any question later.
Thank you so much. Laura Lorenzo, a PhD student from University of Iceland, will close our session. Her study is focused on testing of different drugs, both in the lab and in animals, to determine the best combination of drug delivery system, and administration path to the retina. Her talk will help us to understand the different stages, as well as the importance and challenges of clinical trials.
Okay, thank you, Anna. So following the introduction on clinical trials by Sumaya, I'm going to guide you to the next steps. Next slide, please. So there are 5 well-established phases in clinical trials in order to test each drug, each one of which presents different regulations and objectives. The preclinical phase, as Sumaya mentioned before, follows the phase in which the drug has been developed and includes the testing of the drug in animals. Once this phase is completed successfully, the drug can go into the next phase, which is the first phase of the clinical trials. So here in the first phase, the researchers will test the new drug or treatment in a small group of population that goes from 20 to 80 participants. And here is the first time that they will evaluate its safety in humans. The main objective of this phase is to answer 2 very important questions, which are what is the safe dose of this drug in humans, and does this drug cause any side effects? This stage usually takes from weeks to months to be completed. And once these questions have been answered and the drug is considered safe for humans, it can be taken to the second phase. Next slide, please. So in the second phase, the potential treatment is given to a larger group of people that includes from 100 to 300 participants. The dose and side effects have already been reported in the previous step. So now what is most important to answer is this drug effective once it's administered in a safe dose? Also, the fact that this phase includes a larger group of people will allow the researchers to make sure that this drug is safe for a more general population. Depending on the drug, this stage can take several years to be completed until all information has been collected and it's able to start the third phase. Next slide, please. So in the third phase of the clinical trials, the study drug or the treatment is given to 1,000 to 3,000 people in order to confirm its effectiveness. In this phase, the drug is used in different populations and in different doses to ensure both the safety and the efficacy. And here it is also compared with other common treatments for the same disease, and it's combined with other drugs to study if there are any interactions. If this phase has positive results, then the drug can be approved by the authorities and it will be able to enter the last phase of the clinical trials. In the fourth and last phase, the drug has already been approved for by the authorities. So now it can be tested on thousands of people. The main objectives here will include seeing if less common side effects exist and have not been reported before, and also to evaluate how effective this drug is in relation to its cost. However, even if it takes years to reach this stage, still 4% of the drugs are withdrawn during this phase due to safety issues. Next slide, please. So what does it mean to participate in clinical trials? The participation in trials has both benefits and risks that need to be taken into account before enrolling in one. Starting with the benefits, the positive points for the participants include the access to new and promising treatments that are still not available in the market, the regular medical attention and monitoring to ensure the well-being of each participant, the possibility to contribute in scientific and medical progress and help other people, and the chance to obtain free or low-cost access to new treatments that can otherwise be really expensive. On the other hand, there are also risks that need to be considered, since participating in clinical trial means that the participants are not choosing which treatment they get. So they might be a part of the control group, which will not receive any treatment. The objective of this, as Sumaya mentioned as well, is to make sure that the actual treatment is effective when compared with the placebo group. Also, because we are all different and our bodies respond differently, the drug might not be as effective for a given participant as it was for people who tried it in the previous phases. Besides that, participants can also develop and experience new or more severe side effects that hadn't been reported before. And lastly, what was considered also a benefit, such as getting more medical attention, can also mean an inconvenience for some people, since the need to attend more doctor visits and monitoring in order to ensure that their health is not being compromised can be time-consuming and can interfere with the participants' everyday life. And that's it for my part. If you have any questions.
So the presentations in this session are over. So if you have any question, please raise your hand. There is one question from Raul Manuel Perez Garcia. Could you please unmute yourself and ask the question?
Can you hear me?
Yes, perfectly.
Yes. I was wondering with regards to the molecule that was shown earlier in the talk, could you explain a little bit more in which phase of the clinical trial it is, and which target is it meant to target? Hello?
I assume is the question to Laura.
Me? Yeah, I don't think this drug is in any part of the clinical trials yet. I would say that it's been tested in vitro to test the pharmacokinetics, but yeah, I think that the rest of the question should be answered by Osvaldo maybe, or Dilip, since they are taking care of the synthesis and formulation.
I can add something. It's a common question that I get actually when we're doing this and we're talking about taking this drug to clinical trials and all, and the efforts that we're doing together. But we're not in clinical trials yet. We are in preclinical stages, and I think what we are doing is trying to meet all the requirements to be able to take it to clinical trials. So we haven't done anything on humans. We're still testing on animals and cells, and we're yet to do some toxicology, for example. Yeah.
Great. Thank you.
Thank you, Osvaldo. Thank you, Laura. There's another question from Yet Hilhorst. Could you please ask your question?
First, I want to make my compliments to everybody for the presentations. They give a very nice and clear overview of the whole lengthy process of drug development. And now I'm wondering, for this whole consortium is dedicated to the drug development. For many diseases that's done by the big pharma companies. Are you aware of any other efforts to address this type of disease with a different type of drug? Or is this the only one?
Osvaldo, Dilip, Laura, Somaya.
Maybe I can answer something. So as Lee explained it in her talk, there are different strategies to try to cure these diseases. And one of them is, for example, gene therapy. And now there is on the market already the gene therapy for RP65, which was commercialized by Novartis under the name Luxturna. The problem with this very, so retinal degeneration as I don't remember who mentioned it, but there are more than 270 genes that are mutated. So what we are trying to do in our consortium is to target something that would be common in all these mutations. And this is the cell death pathway. So what Michel explained, the CGMP caused cell death. And the molecule that Osvaldo showed is one molecule that could target this pathway. And if it worked, then it could target the diseases caused by all these genes. And not a gene therapy would target only one type of mutations, for example. And so in the pharma industry, I'm not completely aware of everything that they are doing. I know for sure that there is an ophthalmology department, for example, at Roche or at Novartis, or probably in many other companies. I don't know if they can have a similar approach to ours. I don't know, to be honest.
Somaya?
Yeah.
Is there any other question? Osvaldo, I'm sorry. Please.
No, I had something in mind, but maybe it's the same that Somaya just mentioned. There's a phase 1 clinical trial for retinitis pigmentosa here, ongoing here in Stockholm, Sweden. That's a cell therapy, I think, or gene therapy. So is that the same?
So if it's gene therapy, then I would guess it's targeting one of the mutations in one of the 270 genes, I would say. If it's a cell therapy, I would guess that they try to replace the dying cells by functional ones.
I found it here. It was a gene therapy for RLBP1 mutations, and it is done by Novartis. So I guess it is the one you're talking about.
And I think that Laura, when you came to Tübingen, you saw some gene therapy trials or not?
Yes, I know that there are some trials ongoing. Yeah, I think that they were in an advanced phase. But yeah, I can't really say anything about that.
Is there any other question from the audience?
Actually, I have a question for Dilip.
Yeah.
So Dilip was talking about different criteria for drug delivery and administration. So how important is patient compliance in case of this? So because you were talking about different things like we should consider before the drug delivery. So how important is patient compliance in this scenario?
Yeah, as I mentioned, in case of retinal degenerations, you cannot really put patient comfort at first because there is no approved treatment. apart from the gene therapy, I think it's important to focus on the requirement first. But of course, it's important to consider patient safety. So even we are trying to develop injections. So we are trying to develop injections that are not to be given every often, like every month or every 6 months. So we are trying to increase the drug release duration, by reducing like frequency of injections.
Exactly. Because if we are to give the injection to the patients and it's like very often, maybe the patients will not be willing to take this type of injection because it's too often and it's too uncomfortable or painful for them, right?
Yeah, exactly. That's true.
Thank
But we
you.
haven't really finalized anything about injection or eye drops, or if it is an injection, how frequently we need to give. So everything is in like ongoing.
Yes, thank you.
Thank you, Dilip. Thank you, Manisha. There is another question from Yet Hillhorst. Would you please ask the question?
Yeah, it's a question to Laura.
Mm-hmm.
You mentioned the numbers of participants that you need in the different phases of the clinical trials, and it goes up to thousands.
Mm-hmm.
How difficult would it be to achieve those numbers for retinitis pigmentosa, and can you find ways around it to do those trials with less people?
Yes, of course. I think that that the numbers should be adjusted as to the frequency of the disease or how many people is suffering from it. So yeah, I think that there are ways to go around it for sure. It's under the sponsor or the center providing the treatment to define that.
Maybe I can add also something to it. So what we presented is very general to clinical trials, so very general. And in case of rare diseases, I think things are not happening exactly as we presented them. So for example, when I talk about the placebo, in case of gene therapy, the placebo is not done because if you inject the gene therapy by intravitreal injection, you will induce somehow a damage, we think. Whatever skillful is the surgeon, there will be some damage done in the eye. And then the placebo would be to inject, but then don't inject the active substance. And you need a good reason to inject something in the eye. I mean, I wouldn't go and say, yes, just inject me something, but it's doing nothing to my eye. I don't need it. So you will damage my eye. But at the end, I mean, I'm taking a risk for nothing. So this is ethically not accepted and acceptable. And then, so for these rare diseases, when there is a treatment, I think you would go directly to the patients and skip, for example, the placebos. If there are not enough patients, you would also not meet the requirements in terms of number of people participating.
There's another question from Isaac Shaikh. Would you please ask your question? We don't hear you so well.
Can you hear me now?
A bit better, yes, but I have to admit it can be a bit difficult to understand.
Wait a minute. Maybe now?
Now it's a bit better, yes.
Okay, yeah, thanks for your research. I have one question. In one of the presentations, an organic molecule was showed, a structure of an organic molecule was showed. I think it was Mr. Osvaldo, yeah? So my question is, I didn't get it if you said so, but my question is that drug molecule already in the clinical trial stage, or what's going on with that? Because it's an interesting molecule, as I see.
Yeah, absolutely. So if I understood correctly, you're asking if the molecule that I was showing is in clinical trials. No, it's not in clinical trials. we are working in preclinical phases right now. That means that that is before clinical trials, before doing anything in humans. We are testing everything we can on animals, cells, yeah, to try to meet the demands for going to straight to humans. So we are in preclinical phases.
So another question, how did you know that this drug molecule is going to be helpful in treating the retinitis degenerative diseases? How do you know that? Do you have some results already on some animals? That is the question now. So can I look up in the literature if something is published or patent? Yeah.
We have results, so that's why we are pretty hopeful for this drug. We have results on animals and on cells. We have results of the naked drug as well as the drug in formulations, and there are some papers for that. I think maybe some other of my colleagues can add something. There's a paper by Vicky et al., I think.
Yes, yeah, it was published in PNAS. I don't remember the year, sorry. And the first author is Viggy, and the last author, if I'm not wrong, is François Paquet-Durand. So he's talking about this molecule that Osvaldo presented, and actually our professor started a startup company, and he's looking for investors to start the clinical trial.
Okay, so it means if I just...
It has been tried on mouse retinas, and it has been tried on monkeys as well.
Yes, so if I look for the structure in SciFinder, I should be able to find that published work, I think.
Yes, it's open access as well.
Yeah, I have taken the screenshot. Yeah, thank you very much. Yeah.
Is there any other question?
Hello, I have a question, Fiona here. So I'm wondering how reflective the preclinical stages are when they get to clinical trials. Do you usually see the same success rate? And if not, what is the typical drop-off rate? So how many drug candidates do not make it past the early stages of clinical trials? Is this a lot?
Yeah, there are a lot of drugs failing when they go from preclinical to clinical, just because animal models can be accurate, but they differ substantially from the human being. So yeah, I think that most of the drugs fail in that stage from animal to human.
Can you tell me, like, what are those differences between the animal models and moving to patients?
Not really. I would guess it's about pharmacodynamics, just how the organism absorbs and distributes, and the whole. Yeah, I don't know. I guess it has something to do with that. But
I could add something there, at least an example. So, I mean, the first obvious difference is that an animal will not have the same physiology as a human. For example, if you go from a mouse to a human, the mouse has a really, really tiny eye, and the majority of the eye is just the lens. And so you might not have the exact same flow of the vitreous inside the eye. So the effect of the drug might be different because of that, or it might be different because differences in metabolism. It could be anything.
Do you think that the immune system has an impact? Because you speak about barriers, and this can be like a kind of a barrier for the immune system. Do you think that that has any impact?
I think so. Although I guess that the animal models are selected based on the most similarities for a given disease with the human But yeah, I guess that in some cases that might be the situation. At least that would make sense.
Thank you. There is another question from Raul Manuel Perez Garcia. If you could please ask the question.
Sorry, that was a mistake.
No problem.
Apologize for that.
So, is there anybody having any question? Maybe any of the speakers would like to ask, comment, or just say something? If not, we are actually reaching the end of the conference. So, ladies and gentlemen, I hope all of you enjoyed this short conference, and few hours is definitely not enough to present you all our work and interest in retinal degeneration. But I truly hope we have shown you the challenges, discoveries, and developments of the most important tools to treat retinal disease, and some of which we hope will create meaningful and impactful changes in the lives of those living with those conditions. It was a pleasure for me to be part of this meeting, and I would like to thank all those who have been involved in the organization of this event. Christina Fasser, Fiona Waters, TransMed coordinators, as well as all ESRs presenting and hosting today. Thank you, Manisha, for your collaboration. And then the last information, as Manisha said at the beginning, this event has been recorded and will be available on Retina International website in a few weeks. And if you have any additional questions, do not hesitate to contact us by mail at info@retina-international.org, and we will forward the message to the selected speakers. So thank you so much once again for your attention, and have a lovely Sunday.
Thank you, everyone.